Welcome back. We're on lab 4. These are tissues and the integumentary system. And this isn't a very long lab, but there are a lot of little parts to it that you kind of have to connect. And we're going to look at some slides of different histology of the skin, and and then we'll be done. This will be the end of material for exam 1, or practical, examination 1. And your first exam, you will have 1 week to complete, from Monday morning till Sunday night, because everything's due on Sunday nights. Okay, so we are going to start with introduction to the microscope. Be able to identify the following microscope features: the stage, the lamp, the light source, the objective lenses. We have 4x, 10x, and 40x. Those are scanning low and high magnification levels. We have the coarse adjustment knob. We also have a fine adjustment knob, which is directly over to the right-hand side. We have the arm of the microscope, the nose piece, the base, the stage clips and claw arm, mechanical stage, ocular lens. So we will, there should be a picture in there. Hopefully it got copied over correctly. And we will identify those things. Then we're going to look at the skin anatomy and accessory structures, identifying the layers and the tissues found in our epidermis, dermis, and hypodermis. All right, so the epidermis, the most superficial layer of the skin. Identify the layers and the tissues found in there. So the epidermis, upper layer, and we find stratified squamous epithelial tissue. Stratified squamous epithelial tissue, or epithelium. You can say epithelial tissue or single word epithelium. The dermis, the middle layer, we have dense, irregular connective tissue and simple cuboidal epithelial tissue. Then in the hypodermis, the deepest layer, we have adipose connective tissue and areolar connective tissue. So we're going to look at some of the images, the models of skin. We'll be able to identify the epidermis, dermis, and hypodermis. And that's where I recommend you start. It's like, okay, this is the upper layer, this is the middle layer, this is the lower layer. Because as soon as you see adipose, fat tissue, then you know you're in the hypodermis. So you go, okay, layer, 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 epidermis, dermis, hypodermis. And then you think to yourself, okay, what tissues would I expect to find there? Now, remember, all your tissues, are, either connective tissue, epithelial tissue, nervous tissue, or muscle tissue, right? So epidermis, stratified squamous epithelial tissue. The dermis, dense irregular connective tissue, and simple cuboidal epithelial tissue. And I'll just tell you that all the glands are going to be composed of simple cuboidal epithelial tissue. And so the ones that we see abundantly, the tubes of them, are going to be the sweat glands. And they generally are originating in the dermis and pass through the epidermis to the surface of the body. So we'll see those in some of the models. But we also have oil glands, sebaceous glands, kind of attached to and associated with the hair follicles. And although those actually extend down into the dermis, they're technically part of the epidermis, but we'll often see those in what is the layer of the dermis. Again, I'll show it to you when we see the model. And in the hypodermis, we find those two tissues abundantly: areolar connective tissue and adipose connective tissue. Okay, then we're to identify the structures and functions, found in each layer. All right, so we're going to look at the sweat or sudoriferous glands. What are their functions, or what is its function? 2, that's in the parentheses of the number of functions associated with each. So 2 are with the sweat glands: protection from infection and cools the body, because we know sweat cools the body. But there's components of sweat, the acidity, the kind of salty nature, the hypertonic kind of salty nature to it, has antimicrobial components in it that it's good for protecting against infection. Oil or sebaceous glands protect from infection also, and helps lubricate the epidermis. So that oil, is another antimicrobial type of a substance, but it also keeps your skin from dehydrating. I mean, that's essentially what moisturizer is, some kind of an oil on your skin and keeps your skin moist. Hair follicles, heat retention, protection from UV, and touch reception. The little hairs on your arms and things, when a little fruit fly lands on it, that's touch reception. Then we have 3 types of receptors: free nerve endings, Meissner's corpuscles, and Pacinian corpuscles. Free nerve endings kind of look like a tree. They extend from the dermis into the epidermis. They respond to pain and temperature. So their function is pain and temperature reception. Meissner's corpuscles are pressure reception, or light pressure, light touch. Pacinian corpuscles are also pressure receptors, but they're deeper touch or deeper pressure, because they're found deeper in the skin. And for you to even reach them to stimulate them, because you have to do this physically, then you're going to have to pass through Meissner's corpuscles to even get to the Pacinian corpuscles. So you had to have applied deeper pressure or more significant touch or pressure, to reach the Pacinian corpuscles. So we have light pressure, light touch, deeper pressure, deep touch. Okay, then we're going to move on to the histology of the integumentary system, be able to identify the tissue from slides. For each tissue, identify its skin layer and function, which is supplied. So here we have the tissues that we're going to be able to identify. Right, and here are the layers immediately below in bold print. The layer is either epidermis, dermis, or hypodermis. Okay, we're going to look at the simple cuboidal epithelial tissue. We're going to identify the layer and the function of simple cuboidal, not simple squamous or stratified squamous, simple cuboidal, one single layer of cuboidal cells. Those form glands. Now, it's not the only thing, but the glands in the skin, in the integumentary, it's going to form glands, the sweat glands and the sudoriferous glands. Okay, in what layer? The dermis. Then we have stratified squamous epithelial tissue. Right? That is the epidermis. Right? All that tissue is stratified squamous epithelial tissue. What is its function? Protect from infection, dehydration, and UV radiation. Dense, irregular connective tissue that's found in the dermis. It provides strength with elasticity. And then areolar connective tissue is found in the hypodermis or subcutaneous. Both are terms for the same deepest layer there. It functions to anchor skin to muscle. That's where it's found most abundantly. And then we have adipose connective tissue. That's the fat. That is also found in the hypodermis, or you may refer to it as the subcutaneous. And remember, fats from the second chapter, the chemistry chapter, long-term energy storage, cushion, and insulate organs. So there's not a lot to this, but there's a multitude of pictures that go with these. So let's now change and start looking at our images, because it's only 2 pages, 2.5 pages to fill out. Let's start looking at some images. Right. So here we have layers of, well, you can't see the whole thing, but this is one of those big cookie sheet models. And we've kind of zoomed in on the upper layer. And here where we have this tannish kind of region, which is essentially the keratin in your skin. But the pinkish region here, from the midway here in your epidermis all the way down to the bottom of the pink layer where you see it undulating like waves, that particular region is called the stratum basale. That's your area of high mitotic division. And that whole starting there, the dark pink, the base of the dark pink, all the way up to the top, that is your epidermis. And the tissue is stratified squamous epithelium. Okay, then if we move down a little bit farther, we're kind of zoomed in, so you're not getting, the whole model, but there will be a picture that does allow you to do that. From the pink down to the yellow, right? That middle region where it has that peach kind of background to it, color background to it, right? That is your dermis. What tissues? Dense irregular connective tissue and simple cuboidal epithelial tissue. He left that out. So dense irregular connective tissue and simple cuboidal epithelial tissue, because these yellow tube systems here, which is, they don't have a number. Oh, it's on top, they call it number 21. And at the bottom where this yellow hose kind of looks like, it looks like it's all kinked up on each other, number 22, that's a sweat gland or a sudoriferous gland. And it's composed of simple cuboidal epithelial tissue. We'll look at some of these others while we're here. I'll just say now to the right of that, you can see this brown, or I guess it looks gray here, shaft of something, that's hair. And these glands to the right and left, 10 and 11, and those are the oil glands or sebaceous glands that use the shaft of hair to secrete the oil onto the surface of your skin, thus oily hair. Okay, and then where we have all the yellow and down, that is the deepest layer. That's your hypodermis or your subcutaneous. What tissues? Adipose connective tissue and areolar connective tissues. Okay, so now we're going to look at some of the particular structures that we have wanted you to identify. And one is pointing to this oil gland here, right? This oil gland that surrounds the hair shaft. Okay, so it's a sebaceous or oil gland, keeps the skin soft, it helps to hydrate the skin, protects from bacteria. They're just different ways to kind of describe it. Has antimicrobial properties. Okay, this is one of those Meissner's corpuscles, number 28 here. Kind of looks like a sperm because it has a little tail there, right? Meissner's corpuscles, because they're closer to the surface of the body than Pacinian corpuscles will be, they respond to light pressure or light touch. They are receptors, which means they detect changes like a thermostat in your house. Light touch, light pressure. Okay, then this structure kind of looks like a tree or tree branches. This, excuse me, is a free nerve ending. It begins down here in the dermis and extends into the epidermis. So that's your free nerve ending, and it's a pain and temperature receptor. Pain and temperature. Close to the surface because you want to know if you're putting your hand on something hot. Okay, here we're looking at one of those sweat glands, or sudoriferous glands. Cools the body, acts as an antimicrobial, regulates, body temperature, cools the body. Has antimicrobial functions in it as well. We will look at it again when we look at immunity in the immune system in AMP2. Okay, and then down here, this is a great example of a Pacinian corpuscle, All right. To reach it, you have to go through all the upper layers to get to it. So we say it responds to deep pressure or deep touch reception. The Pacinian corpuscle detects pressure, deep pressure, significant pressure, more than what the Meissner's corpuscles, you're going to get those first on the way through the skin. And then when you also reach the Pacinians, then all this sensory information traveling to your central nervous system is telling your body, that we have, pretty significant pressure on the surface of our body. This is a small model representative of the skin. And now that you saw it on the larger cookie sheet model, you can see what would be the upper layer, right? The epidermis. That would be this entire upper surface, ending in the darker blue region here that's undulating here. Call it Eddie Munster's hairline, because in the Munsters, Eddie Munster had that widow's peak on his forehead. We're kind of old. Okay, we have hair, that acts helps to keep the body warm. It acts as a pressure receptor because light things can land on it, like little gnats or a very light piece of crepe paper or something. Just think of something very light. But it also protects from UV so that your skin doesn't burn. We have the glands that are kind of attached to it and use that. Those would be your oil or sebaceous glands. The white coiled hose here, that would be a sweat gland. Let's see, there are some other, I don't use these for the Meissner's corpuscles, but you can see them here ending just below the darker blue stratum basale layer of your epidermis. Way down here at about, in the hypodermis subcutaneous layer, over to the right-hand side, you can see what's called this lamellated structure. And lamellated means that it's kind of like an onion. It's going to have layers to it. And then the core here is what you want to reach and stimulate that receptor. And that's the deeper one. So that would be a Pacinian corpuscle. Deep touch, deep pressure function. This is kind of an old one, an old model, but it's good for layer. You can see the hair follicle, you can see sebaceous glands and pseudoreferous glands, clearly. You can see a great big Pacinian corpuscle there. Okay, now we're going to start looking at slides of tissues. And we said that we were going to look at, simple cuboidal epithelial tissue by itself. Now, you can often find regions of simple cuboidal on a slide that has, the skin, like an entire specimen of skin that has epidermis, dermis, and hypodermis. It's a little trickier, but sometimes they'll make slides that are very good examples of just that tissue alone. And this is an example of a slide from an area of the body in the kidneys, where we can see a very good example of repeating regions of simple cuboidal epithelial tissue. So these little hoses, or they look like a straw, that is one single layer of cuboidal cells. So simple means one layer, cuboidal is the shape of the cell, epithelial tissue. Simple cuboidal epithelial tissue. They repeat over and over, these little rings. Simple cuboidal epithelial tissue predominates in the dermis. Not that you can't find some in the hypodermis, but they're abundant in the dermis. Okay, then we got to scroll down a little bit. These were pictures taken actually through the eyepiece on the microscope. But this is the skin, and we can see from this pink region here at the tip of the pointer, that is a stratified squamous epithelial tissue. Now above that funny looking substance, it's called keratin, and that's the waxy substance that we have on the palms of our hands and the soles of our feet. We have extra waxy areas there, but the pink tissue there, right through kind of the middle here, and terminating in that, again, that undulating darker pink layers here, kind of look like waves on the ocean, right? That would be your epidermis, and that is stratified squamous epithelium. Right below that dark point, down below that, kind of looks like steak or something here. That is the dermis, right, your middle layer. It's abundant in dense irregular connective tissue, which is most of what you're seeing there. But we also see a good amount of simple cuboidal epithelial tissue because we have so many glands in that region. And then if we go farther down in here, we'd have to go a little bit farther. Once you can see adipose connective tissue, the the fat tissue, then you know you're in the hypodermis or subcutaneous. Okay. So this is a little bit better slide. That epidermis is nice and pink. We go below that. Now we're in the dermis. We can find those two tissues, and we're almost, see down here at the bottom, if you actually scroll down some, just at the base here at 6 o'clock, you can see some simple cuboidal epithelial tissues. This is a sweat gland that's all coiled on itself. I don't think he draws you, or your attention is drawn to it in there, but we can see that dense irregular connective tissue background, and we see examples of simple cuboidal epithelial tissue. Okay, this is another standalone slide, you might call it, as giving you mostly what you're going to see here is adipose connective tissue. It's a very good specimen of fat tissue kind of by itself, but sometimes you'll see some little blood vessels in here. Right, kind of has a honeycomb or chicken wire appearance to it. Large cells with a big fat vacuole in it. So most of what you're looking at is like 90% of the volume of the cell is fat in a vacuole or kind of a vesicle. Right? And then sometimes you can see the nucleus pushed off to the side. And what's taking up stain here is the plasma membrane. The fat, won't take up stain. And often when the specimen is created, the cell gets cut and the fat actually punctures that vacuole or vesicle, and all the fat leaks out anyway. So really all you're going to be able to stain is the plasma membrane. That is adipose connective tissue. It would be found in the hypodermis or subcutaneous. This is areolar connective tissue. So hopefully you looked at some pictures, maybe even in your lecture class. This has this random orientation. It's a loose connective tissue, but it's very irregular, which means that the fibers are going in all different directions. And you can see that these more pink, how to explain this, moderately purple fibers here. They're rather large diameter and rather translucent. Those are the collagen fibers. The darker stainer, narrower fibers here going in all kinds of directions. Like, look, you can see them coursing here. Darker purple compared to these here in a lighter purple, larger diameter. The darker purple ones are the elastin fibers. And areolar connective tissue has both elastic and collagen fibers. Collagen fibers resist over compression and squishing them together. And the elastic fibers allow the tissue, the fiber to be stretched and bounce back like an elastic band. That's why it's called elastin or elastic fibers, found in the hypodermis. And that's it. That is the end of the content. In your lab book, there are pictures of the microscopes. And they label those particular parts of the microscope that we mentioned before. I think previously there might have been a picture that was part of the lab images, and I don't know if it got removed for a particular reason and then never replaced. However, in your lab book, it points to the different parts of the, if you've purchased a lab book, it points to the different parts of the microscope. I don't think I ask questions about it per se, but you should know the parts of that microscope because you need to be able to use the microscopes eventually, in microbiology class. If I can find an extra picture to load up there, which I think would be consistent with what's in the lab book, I will do so.